You see those headlines all the time, right? Scientists find happy people live longer, or this study shows coffee boosts memory. But have you ever really stopped to think, how do they know that? How do they actually get inside our heads to figure this stuff out? Well, today we're going to take that exact journey from a single, simple question all the way to a big conclusion about how our minds work. And this right here is the central mystery. It honestly feels a little bit like a magic trick. I mean, how can a study of, 500 people in Ohio tell us something true about billions of people all over the planet? It's a fair question, but it isn't magic. It's actually a really powerful and precise process, a science all its own. And we're going to break down exactly how they pull it off. So here's our roadmap for this explainer. We'll start with what feels like an impossible task. Then we'll see how researchers find their people, how they paint a picture with the data they collect, and then make that huge leap, the great inference, to a conclusion. And finally, we'll take a quick peek inside a modern researcher's toolkit. First up, the impossible task. Just imagine trying to understand human happiness by asking every single person on Earth. It would be a complete logistical nightmare. It would take centuries, cost an unbelievable amount of money. It's just not going to happen. So researchers had to figure out a much smarter way to do it. And that smarter way is called sampling. Think of it like this. You're cooking a big pot of soup. You don't have to eat the whole pot to know how it tastes, right? You just taste one spoonful. But here's the key. That spoonful has to be a good mix of everything in the pot. In research, the strength of any conclusion depends entirely on how well that small sample really reflects the larger population. Okay, so that brings us to our next big step, finding your people. If that sample is so incredibly important, how do researchers actually decide who gets to be in it? Well, it turns out there are two very different philosophies on how to do this. On one path, you've got probability sampling. This is where every single person in the bigger group has a chance of being selected. This is really the gold standard if your goal is to say something universal. But then there's the other path, non-probability sampling. It's often way easier and faster, but it comes with a serious risk of ending up with a lopsided, biased sample. So with probability sampling, there are a few key ways to do it. Simple random sampling is basically a lottery. Everybody's name goes in a hat and they all have an equal chance. Stratified sampling is a bit more strategic. You might split the population into groups, say by age, and then randomly pick people from each group. That way you make sure everyone's represented. And cluster sampling is all about being efficient. You might randomly pick 10 schools across the country and then study every single student inside those schools. Now for that more practical path. Convenient sampling is just what it sounds like, using people who are easy to get, like college students hanging out on campus. Purposive sampling is when you need specific experts, so you might go out and handpick experienced doctors for a study on hospital stress. And snowball sampling is a pretty clever way to study groups that are hard to find. You find one person and then just ask them to recruit their friends. But, and this is a huge but, all that convenience comes at a very steep price. Something called weird bias. It turns out a massive chunk of what we think we know about human psychology is based on this tiny, very specific slice of humanity, and that creates a giant problem. Can what we learn from a 19-year-old American college student really tell us something fundamental about a 60-year-old farmer in rural Asia? A lot of the time, the answer is no, and that's a major challenge for the whole field. Okay, so you've found your people, you've collected all your information. Now what? Now you're staring at this giant spreadsheet just full of numbers. The next step is to somehow turn that raw, messy data into a clear story. And this is where descriptive statistics come into play. Before we can make any grand claims about the entire world, we first have to simply describe the data we have right in front of us. It's the first step to uncovering the story that's hiding in the numbers, to see the basic patterns within our specific sample. And one of the very first things you do is find the center of your data. The mean is the classic average we all learned in school. The median is the value that's smack dab in the middle, and it's super useful if you have a few extreme scores. For example, if you measure the income of 10 people and one of them is a billionaire, the mean income will be misleadingly high, right? The median gives you a much more realistic picture. And the mode is just the most common value, which is perfect for categories, like finding the most popular choice. All right, now for the moment of truth. This is the big one, the great inference. This is that critical part of the journey where we try to make the leap from our small, measured sample to a big, bold conclusion about everybody. So let's say in your sample of 200 people, you find that group A is a little happier than group B. The million-dollar question is, so what? Is this just some random fluke that happened in your specific group of people? Or does it point to a real, meaningful difference that's actually out there in the world? This is the job of inferential statistics. These are the mathematical tools that let researchers test their ideas and decide if their findings are strong enough to be generalized. It's how they figure out if they found a real effect or if it's just random noise. Now, if you've ever read about a study, you've probably seen this little thing, the p-value. For a really long time, this has kind of been the gatekeeper of scientific discovery. A p-value of less than 0.05 has been the traditional benchmark for a finding to be called statistically significant. But what does that actually mean? It's actually a pretty cool idea. A statistically significant result means that if there were no real effect out in the world, the odds of you finding a pattern this strong in your sample just by pure luck would be less than 5%. It's the researchers' way of saying, hey, we're pretty confident that random chance isn't a good explanation for what we found.
But, and this is so important, significant isn't the whole story. It doesn't mean important. A p-value tells you if an effect is likely real, but it doesn't tell you how big it is. That's the job of something called effect size. For instance, you could find a statistically significant link between eating one extra piece of broccoli a week and having a higher IQ. But if the effect size shows it only boosts your IQ by 0.001 points, is that a meaningful discovery? Probably not.
Okay, to wrap things up, let's take a quick look inside the modern researcher's toolkit. All of this complicated work, from sampling to statistical testing, is powered by some really incredible methods and software these days. And it's really important to remember that not all research is about crunching numbers. Sometimes, the most powerful insights come from simply talking to people. Qualitative analysis dives deep into interviews and written texts to pull out seams and meanings that cold, hard statistics could never find on their own. For the quantitative, number-heavy stuff, the modern workflow usually looks something like this. After they collect the raw data, researchers spend a ton of time just cleaning and organizing it. Only then can they put it into powerful software programs like SPSS, R, or Python. These tools do all the heavy math, but it's always up to the human researcher to figure out what the results truly mean. So the next time you see a headline about some new psychological study, you'll be armed with the right questions. You can ask, who was actually in their sample, and how weird is it? Was the finding statistically significant? And maybe most importantly, how big was the effect? Was it actually meaningful in the real world? Thinking like a researcher is the best way to become a smarter consumer of science. Thanks for coming along on this journey with me.